So in this video, we are going to talk about two techniques, a membrane filter technique and a multiple tube fermentation. And both of these techniques are basically to do water sample testing. And so let's talk about what we're doing. So we'll start with our membrane filter technique. And the purpose of this test is to test a water sample for the presence of coliform bacteria. And this is a presumptive test. So let me give you a few definitions to describe what I mean. So first, let's talk about presumptive. So presumptive means that the results or data has a high confidence above 95% of accuracy, meaning that if we get a positive in this test, we can say with about 95% confidence that in fact the bacteria is coliforms in the water. Now, let's talk about what is a coliform. So in order for the organism to be considered a coliform, it has to meet several criteria. The first is that it needs to be a gram-negative rod. So that's the first criteria. So gram-negative rod. The second part of that is that it ferments lactose. And then the third part is to produce acids and gas. So for an organism to be a coliform, these are gram-negative rod bacteria, 2 that ferments lactose, and 3 to produce acids and gas. So that is how we define what a coliform is. Now, examples of coliform include Escherichia coli, E. coli, Enterobacter aerogenes, Citrobacter fruendii, Klebsiella pneumoniae. These are all examples of coliforms. Now, if you think of E. coli, or you think of Klebsiella pneumoniae, or any of these bacteria, where would you find them? Where is their normal habitat within the human body? And the answer is that those are normally found in the gut. Those are normally found in the gut, right? They're found in the colon. So the question for you then is, why is the presence of coliforms a problem in water? What does that tell you if you see this bacteria in the water? What would be the source of this bacteria likely? And the answer is that coliforms are an indicator of sewage or fecal contamination. So what that tells you is if you detect those coliforms in the water, it's likely that that water is fecally contaminated. Now, that's problematic because if you see E. coli, for example, in the water, that might indicate that other pathogens are also possible in the water. So there could be pathogenic viruses, so in the fecal matter. There could be helminths, worms. There could be the protozoans that we're studying in lab. So the presence of this fecal matter bacteria is problematic because there may be other pathogens in the water as a result. And those other pathogens are much more difficult to test for. Viruses, for example, right? They're obligate intracellular parasites. In order to test if those viruses are in the water, you'd have to culture them within their host cells. Protozoans would be more difficult, worms, etc. So it's much easier to detect the presence of the bacteria in the water and then assume that if we see that fecal contamination, that there's also a possibility for other fecal contaminants as well that might make a patient sick. So what we would be doing in this experiment is that we would be taking our water sample and we would be passing it through a filter. So this is kind of an overview of the setup. Obviously, if we're testing water, it's not going to look this color. But the idea in this case is that you would take whatever water sample. So normally we would have you guys bring in water. So you could test bottled water, for example. You could test sink water. You could test water from a dispenser. Some people like to test like their Hydro Flask water. Basically, you could test, the sky's the limit with what you can test. You could test Huntington Harbor water. You could test any sample that you want. So what you would do is you would have your sample of water, and we have this filter setup. Now, on this filter setup, in this cup that we're going to pour the water into, we have a membrane filter. And so in our example, what we would do for today would be that we would use this nitrocellulose paper. Now let's talk about why we use the nitrocellulose. So this nitrocellulose paper has pores, meaning holes, that are 0.45 micrometers or microns in size, right? So the holes are 0.45, I forgot the 5, 0.45 microns in size. Now, how big are most bacteria? Most bacteria are 1 micrometer to 5 micrometers in size. So 1 to 5 micrometers in size. So you can imagine, let's say this is my pore. This is the size of my hole. If I'm looking at E. coli, for example, which has a size of about 1/2, then the E. coli is going to be too large and it's not going to pass through that pore. It's too big. Even the diameter, let me actually draw that a little bigger. Even the diameter would be too big, and under no way that it turns would it be able to fit through the pores, the openings that are only 0.45 micrometers in size. So if I pour a water sample over a filter, am I going to be testing for bacteria in the filtrate, this bottom part, or am I testing for it on the filter? And so the answer is bacteria would be stuck on filter and would not pass through. So when I pour the water into the collection cup, again, the bacteria is going to get stuck on the filter, and what's going to pass through is not what we're testing. We're going to take that filter, so take that nitrocellulose, and we're then going to put it onto an agar plate and put it in the incubator and look for how much bacteria is on that filter paper, which would tell us how much bacteria was in our water sample. And so this is kind of an overview of what our experiment is going to test. And so this is what we would be doing for this experiment. So this would be our setup for the lab. And so what we would have out is we would have several things. So let me go ahead and put my laser pointer back on. So here is our flask, and the flask would collect the water that goes through the sample. Now, here on the left, here is our water sample that we would be testing. We have a box of nitrocellulose. This has individually wrapped sterile nitrocellulose filters. These are the collection cups, so they detach. So here's our collection cup setup. This is our vacuum apparatus. So you'll notice that if you look underneath these cups, you can see this vacuum setup. And so what happens is when we turn on the vacuum, any water that's in the cup is going to get pulled through, and it's going to, the water has to pass through the nitrocellulose when we set this up, and the water would pass through and the water would end up in the flask. because the vacuum would be pulling or sucking the water through and into the flask. Now, the flask itself, again, we're not testing the water that's in the flask. We're going to be testing for bacteria in the water by looking at the nitrocellulose and basically what bacteria got trapped on the nitrocellulose. We also would have out a beaker of alcohol, and there would be forceps in the alcohol. We would have our Bunsen burners. And so when you do this, you would have your Bunsen burner on, and you would want to make sure that you have your blue inner cone, right? Because that's going to be the hottest part of your flame. And you would need to take your forceps that are dipped in alcohol, and you would flame sterilize them. Now, when you flame sterilize them, right, because they're dipped in alcohol, when I put it in the flame, the tweezers are going to momentarily catch on fire. And that's normal. You wouldn't panic when you saw that. You would just have to wait. Once you flame your tweezers, you would need to let them cool before you touch the nitrocellulose. Now, the nitrocellulose, again, is individually wrapped. So you would open up the package to get the nitrocellulose out. And when you open this package, you would see a white filter that looks like the one in the previous slide. So it has a grid on it. And it would also have a blue backing. The blue backing would not be the part that you want. You want the white nitrocellulose that has the grid. And so when your forceps cool down, once they've been sterilized, then you would touch the forceps and pick up the nitrocellulose. And you would place the nitrocellulose on this little setup. So this little platform here has a place that has pores that will allow the vacuum to pull the water down. So you would have to take your little nitrocellulose and you would place it on here when the cup is not on. So you would place your nitrocellulose on here, grid side up. So you want the bacteria to be on the grid. So grid side up. Then you would take, after you placed it on here, you would take your forceps and put it back in the alcohol. And then you would put the lid, this cap, back on. It's magnetic. So it clips on and it will look like this when it's on there. So you have your nitrocellulose, you put the cup collection part back on top, and then you would pour your water through it and continue on with the process. And so I'm going to walk you through kind of the steps that you would do to do this membrane filter setup. So now I'm going to walk you through how we do this setup. So the pictures that you're seeing on the right are the setup pictures from your lab manual. The ones on the previous slide are what our setup actually looks like for the lab, but the idea is the same. So this would be done in pairs. So per pair, you would test one water sample. And so what you would do is that you would start by taking your forceps. And remember that the forceps are dipped in alcohol. And so you would take those forceps and you would place the forceps in your flame. And again, the forceps are going to kind of catch on fire because the alcohol on the forceps. And so you would take those forceps and let them cool. Now, it's really important that you don't touch the hot forceps to the nitrocellulose. The nitrocellulose is very flammable. It catches on fire very quickly. If you wanted to see this, you can go on YouTube and Google flash paper. And you can see that when you light nitrocellulose on fire, it basically disintegrates very quickly. And so when you're doing this experiment, you have to make sure that your forceps are not too hot, because if so, the nitrocellulose will go up in flames. And so you would have to let your forceps cool. Then you would open the package for the sterile nitrocellulose. So each nitrocellulose filter is individually wrapped. And so you would peel back and open the packaging, and you would use the forceps to grab the nitrocellulose. Now, again, the nitrocellulose is going to be the white part where there's a grid on it. right? So that's the nitrocellulose. In that package is also a blue paper. The blue paper is not the part that you want. That's just in there to kind of protect the nitrocellulose. So you would want to make sure that you're using the white paper with the grid. That's the nitrocellulose. And you would use your forceps to pick up the nitrocellulose and you would put it on the setup grid side up. So you want the grid side up so that when the bacteria goes through, when the water sample passes through, that the bacteria ends up on the grid. So grid side up, put your forceps back into the alcohol. Then you would clamp the filter into place. In our case, it's magnetic, so you would just put the little cup over and it would stick to the base. Now, I'm going to show you this in a minute. So it says dilute the sample according to the lab manual on page 651 or use the water directly. And so what that means is depending on the source of the water, depending on where the water came from, that would dictate if you use the water directly or if you would need to dilute it further. So like if I was testing bottled water, I would be working under the assumption that likely that bottled water should be very clean and therefore I could use it straight without having to dilute it. If I was testing a water sample, let's say that had fecal matter contamination, like we knew that there was a problem with sewage spilling into the water, and I was wanting to test that water, well, then I'm not going to want to use the water straight up. I would want to dilute it first so that I don't get too much bacteria on the grid. So I'll cover that in a minute in terms of the amount of water to use. But basically, you would then take your water and you would pour it into the filter setup. And typically we tell students to pour it to about the 100 milliliter mark. Usually we like to pass 100 milliliters of water through the filter. So you take your water and you're going to pour it into the collection cup. Then after it's in the collection cup, you would turn on the vacuum and the vacuum would pull the water down and into the flask where it would be stored. Now, again, we want the bacteria to be on the filter. So the bacteria would be on this filter here. And so what we would do after we pass the water through is we would turn off the vacuum, and then you take off this collection cup. So take off, disassemble it. Then use your forceps again, and so they're stored in the alcohol. You would need to catch them on fire and then allow them to cool. And then you would use those sterile forceps to then pick up the filter and you would then place it on your endo agar. Now, endo agar is light sensitive. So when you're working with the endo agar, in between, you would want to store your endo agar in your drawer until you are ready to use it. You would not want to leave this sitting out on your desk because if it's sitting out and it's light sensitive, it might affect the results. So we keep them stored in our desk drawer until it's time to do the experiment. And so when I take my nitrocellulose off of this setup, I would then use my forceps and gently lay down the filter onto my endo agar. And you would want to put it so that the agar or the grid side is up, because if you put it grid side down, now the bacteria is trapped between the grid and the agar. So we don't want that. So we want the grid side up. So you use your forceps and you gently just lay the nitrocellulose down onto the agar. And then once it's laying flat, make sure that there's no air bubbles. So you don't want to see these air bubbles underneath. So there's a gap between the nitrocellulose and the agar. It needs to be flat. So you might have to kind of adjust it a little bit just to make sure that there are no air bubbles. Then after you put your nitrocellulose on, you would put your lid back on and then you would incubate agar side up in the incubator for at least 35 degrees Celsius for 24 hours. So our setup is not a one-time use. There are some types of filter setups where the collection cups are disposable. However, that's not the case for ours. So we would have to clean this setup between samples. And so what you would do is you would take that collection cup back off. You would also take this rubber stopper out, and you would rinse both the collection cup and the little stopper setup in or with clean tap water, followed by a rinse with sterile deionized water. So we would have that in a squirt bottle, and we would want to make sure that we rinse the collection cup and the little stopper part that has the filter. You would want to make sure to really rinse those thoroughly to prevent against cross-contamination, meaning to prevent from carryover from one water sample to the other. And so that's how we would do this setup. Now, this slide is basically showing you, depending on where you get your water sample from, how much volume you should use. So for example, again, drinking water. So if you were testing, let's say, bottled water, you would want to use 100 milliliters straight, right? So you would just pour the drinking water into the collection cup. If you're using swimming pool water, probably the same idea, because a lot of times swimming pools might have either chlorine or salt that's there to inhibit microbial growth, and we don't expect to see a lot of fecal contamination. If we're doing wells or lakes, there's a couple different concentrations you could try. You could use just 10 milliliters of the water, 50, or 100. So let's say if we are testing from a lake, for example, and let's say we decided we think that there might be fecal contamination, or quite a bit. So we would use 10 milliliters. Now, 10 milliliters alone is not enough volume to pass through the collection cup. So what you would actually do is, let's say this is my water sample, and this water sample is from a lake. You would actually use a serological pipette to draw up some of the water. And in this case, I would choose 10 milliliters. And we would have out for you these glass bottles. So a glass bottle. And in the glass bottle is 100 milliliters of sterile water. So this water has been autoclaved, it's sterile, and it's basically just there to help dilute out your sample. So I could take 10 milliliters of the water I'm testing, and I could pour it or use the pipette to transfer that water into 100 milliliters of sterile water. Now, when I do that, I've diluted my sample. And that's going to be important because you can calculate the amount of bacteria in the water depending on the amount that you've transferred. We're not going to do that for the purpose of this, and so don't worry about that too much. But the idea is if you're testing, let's say, chlorinated sewage, right? So sewage that has began to be treated. You would use between 1 milliliter, 0.1, 0.01, etc. And so you would need to dilute this first, because if I took this sample and tried to use 100 milliliters of it straight, there's probably so much contamination in there that on the grid, you're not able to see isolated colonies. So that's why we have to dilute it first. So if I'm doing lake again, I would transfer the water from here into the water blank. And I would need to make sure that I mix that water blank really well so that it's evenly distributed. Then there's actually going to be two parts to this that you're going to see. So one part, remember, I talked about that the filter, the membrane filter technique is presumptive, right? It's presumptive. And so that means that tells us that if we get a positive on the endo agar, we can say with about 95% confidence that the organism is a coliform. But 95% is not 100%, right? And so you're going to see in a minute that we will also do what's called a confirmatory test. And the confirmatory test, or the test that will confirm coliforms, is called multiple tube fermentation. And so in a minute, I'm going to talk about this, but we would start with inoculating an LTB broth, lauryl tryptose broth. And so what we would do is when we mix our water in this bottle, before you pour your water through the setup, you would actually want to take some of the water and you would use a transfer pipette, and you would take this water and you would add 1 milliliter to the LTB broth. So you want to use your diluted water to add it to the LTB broth. Now, whatever's left in the bottle is going to go through the filter. So we'll put go through filter, right? So we would pour the water through the filter setup, and then we would take that filter and we would put it on Endo agar. So we would place it on our Endo agar. Now, I'm going to talk about why in just a minute, but a good idea for a control would actually be to repeat this, and then instead of putting the filter on the Endo agar, also put it on nutrient agar. And when I talk about the ingredients that are in the Endo agar, I'll go through why it's useful to have a control that is a nutrient agar. There's a reason, and I'll come back to that. And so this would be our setup for our membrane filter technique. We would need to take some of that water and inoculate an LTB broth. So this is called LTB broth. And then we would pour water through the filter, put the nitrocellulose filter on Endo agar, run the water through another filter, put it on nutrient agar as a control. And again, in a little bit, you'll see why it's important to also include a nutrient agar control. So now we need to talk about our multiple tube fermentation. And so again, the purpose of this multiple tube fermentation is to test a water sample for the presence of coliform bacteria. And again, this is our confirmatory test. So if we get a positive on the Endo agar and we get positive for coliforms on this multiple tube fermentation, well, then we can say with high confidence that in fact there are coliforms in that water sample. So notice the name is multiple tube fermentation, which means that this actually is going to span several days. There's several tubes that you have to inoculate in order for this test to be in fact confirmatory. So day 1, the first tube is going to contain lauryl tryptose broth, which we abbreviate LTB. Now, for our lauryl tryptose broth, the first ingredient in there are the tryptose or the peptones. And so when we talk about media having peptones in it, recall what are the peptones in the media for? What is their purpose? And that is general growth. They are just food for general growth. Next, we have lactose. Lactose is there for fermentation, because remember what we're trying to do in this experiment is to look for coliforms. And so when we look for coliforms, remember that they would be gram-negative bacteria that ferments lactose to produce acids and gas. So we need to have lactose there in order to detect coliforms. So the lactose, again, is going to be the substrate that's going to allow us to detect coliforms. Now, the next ingredient that's in the media is sodium lauryl sulfate. Sodium lauryl sulfate you might see in shampoos as an ingredient. Sodium lauryl sulfate is a detergent. And its purpose in the media is that it inhibits gram-positive growth, but allows for gram-negative growth. So it is going to inhibit gram-positive growth, but allow for gram-negative growth. Now, does that make sense to you? Remember that in this test, we're trying to detect coliforms. Coliforms are gram-negative rod bacteria that ferment lactose to produce acids and gas. So what we're trying to do is we want to encourage the growth of gram-negative bacteria, but inhibit gram-positive. And so the first ingredient, the sodium lauryl sulfate, is going to help us accomplish that, because we should see in this media that the only thing that should grow should be gram-negative, because gram-positives are being inhibited. And then lastly, we have our Durham tube. Why would our tube have a Durham tube? What is the Durham tube used for? And the Durham tube is used for to look for gas production. Because again, a coliform is a gram-negative rod bacteria that ferments lactose to produce acids and gas. So here we're detecting the presence of the gas. We're seeing if they can produce that gas. So you'll notice that now the shift of this, this section of the class is that it's not going to be your biochemical sheets. So in this section, it's not substrate, enzyme, products, color change, etc. When you're studying for these types of tests that we're going to be looking at, you're going to want to study what are the ingredients in the media that are responsible for the result. And so we talked about this in chapter 6. And in chapter 6, we looked at different types of media. And so we talked about reducing media. And in one part, we talked about selective and differential media. And so selective media, notice, says inhibits certain types of organisms from growing while allowing the growth of other types. So what that basically means is, in our example for LTB, our selective ingredient is our laurel. So that would be our laurel, YL. Let me erase that whole thing. Our sodium lauryl sulfate. Sodium lauryl sulfate, because it inhibits gram-positive but allows gram-negative growth. Pen's getting a little crazy here. And so that ingredient in the media we would call selective, because the sodium lauryl sulfate is going to inhibit gram-positive but allows for gram-negative growth. And again, that makes sense because in our coliform test, we're trying to detect gram-negative bacteria. Differential media is used to differentiate closely related bacterial species. For example, by changes in color based on differences in bacterial metabolism. So sometimes it's that you're going to see a color change, which tells us that the organism is using that food source. In our example, so in our LTB, our differential ingredient is going to be our lactose. Because we want to see, so determine if fermented. Because again, we're trying to separate coliforms from non-coliforms based on differences in lactose metabolism. Because you're going to have some gram-negative bacteria that are not coliforms, meaning they'll grow in the LTB, but they won't ferment the lactose. And so lactose is going to be our differential ingredient. It's going to allow us to differentiate between coliform bacteria that ferment it and non-coliform bacteria that do not. In the case of our LTB, that differential ingredient, that lactose, the way that we're detecting it, detect by gas in the Durham tube. So in this example, we're not looking for a color change per se. Instead, we're looking at to see if there's gas in the Durham tube. But in many other cases, you're going to see that we're actually looking for a color change. But for this one, we're looking for gas. So for the multiple tube fermentations, this would be done in your groups of 4. And so what we would give you is unknown A and unknown B. And so what that means is unknown A is one water sample and unknown B is another water sample. And so when we do this in our class, we do it kind of crude. It's not very quantitative. And so you would just take one loop full using a loop. And so what that means is you would get that unknown A water and you would flame sterilize your loop, let it cool, and then dip that loop into the water and transfer some water into that LTB broth. And you would do that for both unknown A and unknown B. Now remember that the endo agar and the membrane filter technique is done in pairs. So each pair has their own water sample. So in a group of 4, you would have 2 water samples that you're testing. One pair would test one and the other pair would test another. And so what you would do is for this one, you would transfer 1 milliliter using a serological pipette. So again, that goes back to the earlier diagram where I showed you that when you dilute the water, you take out 1 milliliter and you put that into the LTB broth. And so you make that transfer using your serological pipette. You and your pair would do that, you and your partner, and the other partner, the other pair in your group of 4 would do their water sample. So you would end up with 4 LTB broths per group of 4. You would take your 4 tubes with your LTB broth, you would put them into our can, and then you would put your can in the incubator and incubate at 37 degrees Celsius. And in our case, we do this for 48 hours. So now we're going to move on to day 2, meaning what we would do on the readout 48 hours after our incubation. And so this would now be what we would do on the following class period. So now we would look at our results for our membrane filter technique. And remember that the purpose of our membrane filter technique is to test a water sample for the presence of coliform bacteria. And again, this is a presumptive test. So the endo agar alone is presumptive. It's not going to be able to tell us with 100% certainty whether or not coliforms are in the water, but it does give us about 95% confidence. So what is in that endo agar? Be aware that the recipe for the endo agar in the book is a little bit different than our endo agar. In the endo agar in the book uses sodium deoxycholate and sodium lauryl sulfate. That's actually not in our endo agar. These are the ingredients that are listed here that are in our agar. And so again, this is going to be a little bit different, but the idea is going to be the same. So let's talk about what's in the endo agar. So the first ingredient that's in there is the yeast extract and the peptones. And so what would the yeast extract and the peptones be used for? And that is, it's food for general growth. Food for general growth. Now, next we have lactose. What is lactose in there for? And it's there for fermentation. Because we're going to be able to detect coliforms. Because coliforms metabolize lactose to produce acids and gas. So lactose is there because it's going to allow us to distinguish between coliforms and non-coliforms. So next we're going to have our dipotassium phosphate and monopotassium phosphate. These are our buffers used to keep pH constant. Because again, cells can't tolerate big swings in pH. So we need buffers in there to keep the pH relatively constant. The next ingredient that's going to be in there is going to be our sodium sulfite. And our sodium sulfite is going to darken with acetaldehyde, which is going to detect coliforms. Now, acetaldehyde, you might recall if you watched the metabolism video, the acetaldehyde acts as the final electron acceptor during alcoholic fermentation. And so during alcoholic fermentation, acetaldehyde is going to be our final electron acceptor. So if the lactose is metabolized, right, if the lactose is used as a food source and it's fermented, the lactose is going to get broken down into glucose. Glucose is going to get broken down into pyruvate. Pyruvate is going to be decarboxylated. It's going to remove a CO2 and it forms acetaldehyde. So if there's acetaldehyde in the media, the colonies are going to darken. And that is used to detect coliform. Because what that tells us is that lactose was metabolized. Because again, the only way that you get darker colonies is if the lactose was metabolized. Because if it was, it produces acetaldehyde and the sodium sulfite will darken with the acetaldehyde. The other reason that it's in there is that it's used to inhibit gram-positive bacteria, but allow gram-negative growth. And again, that makes sense because if you think about it, right, we're looking for coliforms, which are gram-negative rod bacteria that ferments lactose to produce acids and gas. And so this ingredient, the sodium sulfite, would basically inhibit gram-positive growth. Sulfites are added to red wine often to prevent spoilage because it inhibits gram-positive bacteria. And so again, in a minute, I have a slide to show you about the sodium sulfite and the acetaldehyde part. But again, if you've watched the metabolism video, you've already heard about acetaldehyde. The next ingredient is the basic fuchsin. And so the basic fuchsin is a basic dye, and it also inhibits gram-positive growth. So again, it's going to inhibit gram-positive but allow for gram-negative. Now, in addition to inhibiting gram-positive, there's another reason that basic fuchsin is in the media. And so if it turns a metallic green sheen, it means that there's a decrease in pH because of increase in acid. And what that tells us is that the bacteria are rapid fermenters of lactose. Because remember that the product of sugar metabolism are acidic. And so if the organism is able to rapidly ferment the lactose, it's going to produce a lot of acids. And when it produces a lot of acids, that's going to drop the pH. And the basic fuchsin, this dye, is going to turn metallic green sheen when the pH decreases. Now, if you see that the colonies are dark pink or dark red, this means that there's no change in pH and that they are slow fermenters. So let me explain that. So they are slow fermenters. So remember that if you look at the list of ingredients that are in the Endo agar, any bacteria that grows on the plate, what conclusion can you make if you see growth on the plate, no matter what color it is? If you see growth, what does that tell you about that bacteria that grew on the plate? And the answer is that if you see growth, it tells you that there are gram-negative bacteria in the media. So that would include our coliforms. This would include our non-coliforms, right? What that means if bacteria grows on this plate is that it means that the organism is gram-negative. So if we look at our ingredients, which of our ingredients that are in here would be selective ingredients? And the answer is the sodium sulfite is selective, and the basic fuchsin is selective, because those two ingredients are going to inhibit gram-positive growth but encourage gram-negative. So we have our selective ingredients. Now, on our plate, we're going to have all gram-negative bacteria, but we need a way to distinguish coliforms from non-coliforms, because not all gram-negative bacteria would be considered a coliform. So we, in addition to having selective ingredients, we also have differential ingredients, right? And so what would be our differential ingredient? What are we trying to determine if they metabolize to determine if they're a coliform? And the answer is my differential ingredient would be the lactose. That's how we're distinguishing between coliforms and non-coliforms, because on our plate, we have all gram-negative growth, but we need to still know if they are coliforms. And so our differential ingredient that's in the media is going to be the lactose. Now, anything that grows, right, is gram-negative, but we need to be able to distinguish coliforms from non-coliforms. So if the organism utilizes the lactose, remember that they're going to produce the acetaldehyde. The sodium sulfite is going to darken with acetaldehyde. So if lactose is fermented, the sodium sulfite is going to darken with acetaldehyde, and you end up with colonies that are dark in color. So any dark in color colonies would be coliforms. But we can take it a step further and also look at the change in pH. And the change in pH comes into play with the basic fusion. And that is that it allows us to distinguish between rapid fermenters like E. coli, versus slow fermenters, which would be the case for Klebsiella and ammoniae. So this is just an added layer, this basic fusion, in addition to being selective. It also basically helps us to distinguish between the rapid fermenters from the non-rapid fermenters. If the colonies darken, we know that the lactose was fermented. But the basic fusion is going to help us determine if it was fermented rapidly or whether it was fermented slowly. Now, if it's a slow fermentation, that doesn't mean that acids won't be produced. It just means that it's going to take longer. And so you're not going to be able to see that change within the time frame that we test it. So basically, this is just showing you kind of how the sodium sulfite works. So lactose is a disaccharide. It gets metabolized. It gets broken down into glucose plus galactose. The glucose can go through glycolysis. So it's going to break that 6-carbon sugar in half to 2 3-carbon pyruvates. When we break that apart, we're going to transfer electrons onto an electron carrier. So we have our NAD+, and it's going to gain electrons, and it's going to become NADH. Again, this goes back to the metabolism lecture. We also get a little bit of ATP out. We get 2 ATP per glucose by doing glycolysis. If O2 is not available, right, or if the bacteria does not do aerobic respiration, then they can go through, and this diagram is showing alcoholic fermentation. And so what's going to happen is that the pyruvate is going to break apart, and for each pyruvate, we get 1 CO2. We have 2 pyruvates, so we get 2 CO2s. The remaining 2-carbon part is our acetaldehyde. This is our final electron acceptor. And again, it's going to darken with sodium sulfite. So if our colonies are dark, right? If our colonies are dark, that tells us that the lactose was metabolized. Because again, the only way you get colonies that darken is if the bacteria use the sugar. Notice in the media, was there any glucose? Answer is no. That was done intentionally, right? Because if given the choice and bacteria can choose between lactose and glucose, what are they going to choose? They're going to choose glucose. And so, but we need to know specifically for lactose. So the only sugar that's in there is lactose. If our colonies are dark, that tells us that the lactose was metabolized. Now, we're not only going to see dark colonies on our plate, you would also likely see light colonies, meaning that they have not darkened. And that's going to tell us something different. And I'll get to that in just a moment. So this is showing you some examples of what this might look like on a nitrocellulose filter. So this over here is our metallic green sheen. So it's important that when you look at these, that you would look at the nitrocellulose itself. So look at the bacterial colonies on the filter and not on the agar that surrounds it. Because sometimes what you'll see is that the agar around it has this metallic green sheen, but we're actually looking for the colonies on the nitrocellulose because that's where the bacteria, or that's where the water sample passed. And so we're looking for bacteria on that filter. This bottom picture has two types. We have our light pink, and the light pink is also colorless, meaning it does not darken. And then we have our dark pink or red. And so we have different interpretations depending on the types of colonies that we see. Now, when we test our water sample, there could be a mix. You might see some dark colonies. You might see some light, right? You might see some variation. But let's talk about what the results mean if we get these types of growth patterns. So let's start with the metallic green sheen. So first things first, it grew. What does that tell us about the bacteria on the plate if it grew? So what does it tell us about the bacteria if you see colonies that are metallic green sheen? And the first part is if it grew, you know it's gram-negative because we have selective ingredients that inhibit the gram-positive but allow for gram-negative growth. So first conclusion we can say is if it grew and it's metallic green sheen, it grew, so that tells us that that bacteria is definitely gram-negative. Now, next part, the colonies darkened. Right? The colonies darkened. They're not light in color. So what that tells us is that if the colonies have darkened, that acetaldehyde was produced and it reacted with the sodium sulfite to cause the colonies to darken. The way that we get acetaldehyde is if the lactose was fermented. So that tells us that the lactose was fermented. If they're metallic green sheen, we can take that a step further. Metallic green sheen, again, means that the pH has decreased because there are acids that were produced. So this would be a rapid fermenter of lactose. So again, grew, so it tells us gram-negative, darkened with acetaldehyde, and turned metallic green with the low pH. So this organism would be a rapid fermenter of lactose. Our identification based on these metallic green sheen colonies is that this is presumptive Escherichia coli, presumptive E. coli. E. coli is responsible for a variety of different conditions. 90% of all urinary tract infections, UTIs, are caused by E. coli. E. coli can cause food poisoning, right? So if you're looking at the O157H7, that can be caused by E. coli. So there are a lot of diseases that go with E. coli. But if we got this result, would that be considered positive for coliforms? So would this bacteria be coliforms? And the answer is yes. This is positive. And it's positive because it's a gram-negative bacteria that ferments lactose to produce acids and gas. Notice in this one, we're detecting the acids, but in this example, we're not detecting the gas. The multiple tube fermentation is used to detect the gas part of this. So this is only presumptive though. So that's the metallic green sheen. That would be considered a coliform. So now let's talk about the dark pink red. So if the colonies darkened. So it grew, right? And if it grew, what does that tell you? It tells you that it's gram-negative. The colonies darkened, right, with acetaldehyde, but did not turn the metallic green sheen, which means that there were not enough acids there to cause it to become green, which tells us that that is a slow fermenter of lactose. It did still, in fact, use the lactose because the colonies darkened, but it did not produce enough acids to cause the metallic green sheen. And so if that's the case, right, they are slow fermenters of lactose, and that would be presumptive Klebsiella pneumoniae. And so Klebsiella pneumoniae would be the organism that would likely be. Can't say with 100% certainty, but it's presumptive Klebsiella pneumoniae. And Klebsiella pneumoniae is normally found in the gut. If it gets into the lungs, however, it causes pneumonia. So would dark pink red, would that be considered positive or negative for coliforms? And the answer is that that's considered positive because it is still a gram-negative bacteria. It does ferment the lactose because the colony is darkened. It just did not produce enough acids to be detected by the basic fusion. But the fact that they darkened and the fact that they grew, we're going to call that positive for being a coliform. Now, let's come to our last example, which is the light pink or colorless. And what it means by colorless, not that the colonies are clear, but that they just look like the agar itself. So, light pink and colorless tells you it grew, right? So it's a gram-negative. It did not darken, so was there acetaldehyde? And the answer is no, right? Because if acetaldehyde was there, the colonies would have darkened. Notice that the bacteria grew. What did the light pink bacteria use as a food source? What is the other food source in the media that it would have used? And the answer is peptones, right? So if the colonies are light pink, the bacteria did not ferment the lactose. Used peptones instead. So for this one, right, the presumptive, so let me put this up here for you. So grew, it's gram-negative, did not darken with the acetaldehyde, which means it did not ferment the lactose, but instead used the peptones. This is presumptive Salmonella typhi, which causes typhoid fever, or Shigella dysenteriae, which causes shigellosis. So let me just kind of briefly talk about these two diseases. So if you think about Salmonella typhi, how many people have heard of Typhoid Mary? Well, Typhoid Mary was a chef in the 1900s and got a mild case of typhoid fever. This has flu-like symptoms, and then she felt better and she went back to work. When you get Salmonella typhi, the bacteria is in the fecal matter, and during this time, toilet paper was not commonplace, nor was necessarily washing your hands after going to the restroom. And so she didn't have symptoms of typhoid fever, but she still had the bacteria. And because she was a chef, she went back to preparing people's food, right? And so she herself still carried typhoid fever. And so she would go to the bathroom, end up with the bacteria on her hands, and then go and prepare food, and people would get sick with typhoid fever. So she was infecting people. However, she did not believe that she was infecting people because in her mind, she wasn't sick. So how could she be the cause of other people getting sick? And so at some point, the government actually isolated her on an island in New York, but eventually was released and allowed to come back, but was told that she could no longer work as a chef. However, she didn't know really what else to do, and so she did eventually go back to working as a chef. And then more people ended up with typhoid fever as a result. And so overall, Typhoid Mary was responsible for about 47 people contracting typhoid fever and 3 deaths linked to typhoid fever. So she was the super spreader of typhoid fever. And that's because she was a chef and she was not washing her hands after going to the bathroom and was spreading the bacteria to other people whose food she was preparing. In terms of Shigella dysenteriae, notice the dysenteriae. Dysentery should now be familiar to you because you learned about that term in the protozoan lecture. And so dysentery is an inflammation of the colon, and it causes diarrheal disease. And so when people get shigellosis, it's basically going to mimic food poisoning. So upset stomach, diarrhea, maybe vomiting, etc. And shigellosis is problematic in poor sanitation areas. So areas where fecal matter is not properly disposed of, where people might not have access to clean water. You might remember that within the top 10 causes of death in the world, so not the US, but in the world, top 10 causes of death includes diarrheal disease. Now, again, we don't see that much in the US, but in other countries, diarrheal diseases actually are a major cause of death because of severe dehydration. And so shigellosis is particularly bad in places with poor sanitation. And so that's where you could typically contract Shigella dysenteriae. The E. coli O157H7, right? The strain of E. coli that makes you sick with food poisoning. It causes disease unlike normal E. coli in your gut because that strain of E. coli, that O157H7, acquired the Shiga toxin gene from Shigella. And so that's why the symptoms of that E. coli are going to be similar to the symptoms of shigellosis because it's the Shiga toxin produced by Shigella. So basically, if we're looking at light pink colonies, light pink colonies, they grew, so they're gram-negative. They did not darken with acetaldehyde. They did not ferment lactose. Is that positive or negative for coliforms? Would we call that bacteria coliforms? Answer is, it's considered negative, right? It's negative because it did not ferment the lactose. And in order for it to be a coliform, it has to be a gram-negative rod that ferments lactose to produce acids and gas. So this bacteria would not be a coliform. It is a gram-negative bacteria, but it's not a coliform. Now, another example of a bacteria that we've talked about a lot that might produce light pink colonies, Alkaligenes faecalis. Alkaligenes faecalis, remember, does alkaligenes use sugar? And the answer is no. It's non-saccharolytic. It evolved in the gut alongside E. coli, and it only uses peptones as food. So alkaligenes would basically also appear light pink on this grid. So this is basically showing you different bacteria and what it looks like when grown on this nitrocellulose. So we have over here on the far left, the Klebsiella pneumoniae. You can see that the colonies have darkened. So they're the dark pink red. Again, this is positive for being a coliform. We have E. coli, which has a metallic green sheen. It darkened. It's positive. And then lastly, we have Alkaligenes faecalis. If you look closely, you can see these little tiny dots in there, but the dots are colorless. They only appear the light pink because of the endo agar itself. So Alkaligenes faecalis would be negative. It's not a coliform. It is a gram-negative bacteria, but it's not a coliform. So now that we've kind of talked about the readout, I want to go back and address why using a nutrient agar. So why using a nutrient agar? And in fact, there's a question in your question set. It's actually under the McConkey agar part on your question set. And if you're looking at your question sets, you would want to turn to question 137. And question 137 says, explain what the results on the nutrient agar control plate prove in this experiment. So this interpretation for a nutrient agar, the Na is nutrient agar, the nutrient agar control plate for this would be the same as the McConkey. The McConkey agar is also used for coliforms. You will not be tested on the McConkey agar on the lab practical. However, you do need to answer those question sets because that information about the McConkey agar is going to be used for your unknown project. And so you do need to review that. It does go with our coliform testing. So you want to answer those questions. But again, they're not on your practical. But this question does relate to endo agar, so I'm going to address it here with you. So, explain what the results on nutrient agar control plate prove in this experiment. So again, I told you to pour your water through the filter twice, 2 different filters. One filter would go on endo agar, and one filter would go on nutrient agar. So, first thing I want to ask you is if I take that filter and I pour the water through it and I put my filter on endo agar, right? So I put my filter on the endo agar. So let me use my laser pointer, right? So I take my filter, I put it on the endo agar. If I get no growth on endo agar, so let's say there's no colonies on this filter paper. If there are no colonies, can I conclude that there's no bacteria in our water sample, right? So if I run this through the filter and I put it on endo agar and I see no bacteria, can I conclude that there's no bacteria in the water? And the answer is no. I can't make that conclusion. Because remember that this media is selective. The only thing that can grow on it is the gram negative. So even if I had gram positive bacteria in that water sample, if I had that gram positive bacteria, right? If I had that gram positive, it's not going to grow on endo agar. But if I run the water sample through another filter and I now put that filter on nutrient agar, nutrient agar is used to grow a wide variety of bacteria. Gram positive, gram negative can all grow. If I use a nutrient agar as a control and I put a filter on there and I look at the filter 48 hours after incubation and I see no colonies on there, then I can say that there was no bacteria in the water. So it acts as a point of comparison to determine any bacterial growth because the endo agar is only going to tell you about gram negative bacteria in the water. Because again, in the endo agar, we're looking for coliforms specifically. Nutrient agar control acts as a control to look at all microbial growth. So we're not just looking at gram positive, we're looking at gram positive and gram negative, right? And so that would be your answer for number 137, is that it is a control to look for all microbial growth. And so here are some examples of water samples that we have tested in the past. Huntington Harbor water, for example, will sometimes have coliforms. So look at the one on the left. You can see a variety of different colonies. Some have that kind of metallic green sheen. Some are the dark red. Some are the colorless. So that means in this water sample, we have a variety of bacteria, some of which are coliforms, right? And so the darker colonies indicate that those are coliforms. And some of the bacteria are non-coliforms, right? They're light pink. If I look at the one on the right, notice that the concentration of bacteria was so high. See how you see this like dark ring here? That means that this whole area is covered in bacteria. And there's no really way to quantitate your number of colonies if it looks like this. The reason that it has a grid on the nitrocellulose is that one of the things you have to be aware about is that drinking water to be considered safe doesn't have to have zero bacteria. There is an acceptable amount of bacteria that is considered acceptable. And so it depends on how many colonies you get per 100 milliliters of water. So if you have one colony per 100 milliliters of water, that's considered an acceptable amount of coliforms. But above that would not. And so by having these colonies here, it would allow us to quantitate how much bacteria was in that water to see if that water would be considered safe to drink. And so if you don't dilute it enough, you end up with this basically lawn of so much bacteria that you don't see all those individual colonies. So the one on the left is a little bit better. Now, in terms of our experiment, so the way we set up our experiment has a caveat. And that is that we use those collection cups where we pour the water in over and over and over again, meaning that we do clean them out in between water samples. But sometimes what happens is we do actually get carryover from one water sample to another. And one of the ways that we can distinguish that possibility is by also looking at our LTB broth. Because if we see colonies on our endo agar, we would also expect to see growth in the LTB broth. Because remember, those came from the same water sample. And so oftentimes what happens when we do this experiment is that students will get colonies on their endo agar plate. They'll end up with some colonies. But when they do their LTB, they don't get any growth. And what that interpretation would tell us is that if you don't get any growth in the LTB, it's probably likely that any growth that's on your filter is carryover from somebody else's water sample. You can see this one here. See how this part out here is clear? There's no colonies out here. That's where the collection cup sits on top of the filter. See how you can see these colonies on that part? What that means is that because that part was normally in the magnetic part where the cup sits, those little colonies there are likely from the collection cup itself, meaning that it carried over from the previous water sample. So again, they do have disposable one-time use type setups for membrane filtration, but our setup doesn't do that. We use the same setup over and over with new nitrocellulose, but you could get carryover from one water sample to another. And that's why having the multiple tube fermentation is also important as a confirmatory test, because this says there could be, right? It's presumptive positive, but we would need the multiple tube fermentation in order to confirm it. And so this is our endo agar. And so membrane filter and endo agar go together. Those two tests go hand in hand. So now back to our multiple tube fermentation. Again, purpose of our multiple tube fermentation, to test a water sample for the presence of coliform bacteria. This whole test, so all the tubes, would be confirmatory. The first test that we set up as part of our multiple tube fermentation was the LTB broth. And again, the LTB stands for lauryl tryptose broth. And just a quick reminder as to what was in the media. We have tryptose, which is our peptones, food for general growth. Lactose is our substrate for fermentation. It's there to detect coliforms because coliforms should be able to ferment the lactose to produce acids and gas. We have sodium lauryl sulfate. Sodium lauryl sulfate is a detergent. Remember that that is our selective ingredient. It's going to select for gram-negative growth, but inhibit gram-positive, right? And so that ingredient is selective because we're looking for coliforms, which are gram-negative. And then lastly, we have our Durham tube. And our Durham tube, remember, is to detect the presence of gas. So now let's talk about what our results would look like in the LTB broth. So there are a couple possibilities. So here on the left, right, this tube has no growth and no gas. So let's say we had added water to this and we got that result, right? No growth and no gas. Does that mean that our water has no bacteria? Answer is no, it does not mean that it has no bacteria, because again, the only thing that can grow is gram-negative. So what that tells us is that there's no gram-negative bacteria in the water, right? So no turbidity. That's one result. And so is that positive or negative for coliforms? And the answer is that is negative for coliforms because there's no gram-negative bacteria. But there's another negative for the LTB broth, and that is turbidity. Turbidity with no gas. Turbidity with no gas. What that means is that the bacteria is able to grow, right? It was able to grow, which means it is gram-negative bacteria. But is it a coliform? And the answer is no, but not a coliform. So again, this is a negative because a coliform needs to ferment lactose to produce acids and gas. So if we get turbidity with no gas, turbidity means it's gram-negative, but no gas means that it did not ferment the lactose to produce acids and gas. That's a negative. Our positive in the LTB, so our positive is going to be turbidity and gas. Because what that means is that it's gram-negative and ferments lactose. So if it grew, right, if it has turbidity and it has gas, what that tells us is it's gram-negative because it grew and it was able to ferment lactose to produce gas. So notice that the Endo agar allows us to detect the acids. The LTB allows us to detect the presence of the gas. And so if it grew and it has gas, that would be a positive in the LTB. Now, the LTB alone, if you didn't continue on, the LTB alone would still be considered presumptive. And that's because you have to think about in the LTB, there's only one selective ingredient. And the selective ingredient in that one is the sodium lauryl sulfate. Now, you can imagine that it's possible that while sodium lauryl sulfate should inhibit gram-positive and only allow gram-negative growth, it is possible that bacteria could be resistant to the sodium lauryl sulfate, meaning it doesn't actually inhibit them. So we need to have a follow-up test that uses other types of selective ingredients in order to confirm this. And so let's start by talking about what is in the next step. So the day 2, you would inoculate what's called a BGLB, Brilliant Green Lactose Bile Broth. So that's the name of the media. So first ingredient that we need to know is going to be our peptones. And what would the peptones be in the media for? General growth. So this is our food for general growth. So that's our peptones. Next, we have lactose. Lactose is there for fermentation, to detect coliforms. Because again, in order for the bacteria to be considered coliforms, they need to be gram negative. They need to ferment the lactose to produce acids and gas. So we need the lactose there as our differential ingredient, right? This is our differential ingredient. So lactose is there for fermentation to detect coliforms. Next, we have oxgal. Oxgal is referring to the gallbladder of an ox. So if you know anything about the gallbladder is going to be the source of bile. So let me start by explaining that part. So your body produces bile by the liver, and it's stored in the gallbladder. When you eat fatty foods, the gallbladder is going to take that bile and it's going to put it in the intestine. And the bile is there to serve as what we call an emulsifying agent. And what that means is that bile's job in the digestion of fats is to emulsify the fats, make it more likely to interact with the water, because normally oil and water don't mix. And so what happens is that the bile is there as an emulsifying agent to help mix the water and the fat. And so you can think of an analogy for how bile works. Bile doesn't actually break down fats. It helps prepare the fats. And so think of it like the fat is like a ball of cookie dough, right? If I want to cut out cookies from my cookie dough, if I want to cut that dough, just like if I wanted to break down lipids. So if I want to cut my cookie dough, can I leave it as a ball? And the answer is no. If I want to be efficient in my cutting, I need to roll out the dough. I need to use a rolling pin and flatten out the dough, increase the surface area so that the enzyme lipase can come in and cut the fat, or so that I can roll it out and I can cut out my cookie shapes. So bile, think of like the rolling pin. Its job is to roll out or increase the surface area of the fats to help get the fat ready for lipase to break down the fat. So bile is basically an emulsifying agent that helps your body with the breakdown of fats. But in this experiment, bile does something a little bit different. And so the oxgal is the gallbladder of an ox, which means that it has bile in it. And the purpose of this ingredient is that it's there to inhibit gram-positive, but allow gram-negative growth, right? So it's there to inhibit gram-positive, but allow for gram-negative growth. What would you call that type of ingredient? And that ingredient would be considered selective. And then lastly, we have our last ingredient, which is our brilliant green dye. And the brilliant green dye is also there to inhibit gram-positive, but allow gram-negative. Because again, we're looking for coliforms. So we would also call the brilliant green dye selective. That ingredient is selective. It's selecting for gram-negative growth. So notice that in the LTB, the selective ingredient was the sodium lauryl sulfate. In our confirmatory test, in our BGLB, we have more selective ingredients. Now we have our oxgal or our bile, and we have our brilliant green dye. If bacteria grows in both sets of tubes, if it grows in the LTB and it grows in BGLB, it's not likely that it's not likely that there's a gram-positive bacteria that is resistant to all three. So by having this different combinations of selective ingredients, it helps us to confirm that in fact anything that grows in there is gram-negative. So what you would do for this is any LTB broth that was considered positive, meaning it had growth and it had gas. The gas is CO2, right? So if you remember back to the lactose fermentation, when we go from pyruvate to acetaldehyde, CO2 is produced. So the gas that's in the durum tube is that CO2. You get that gas if the lactose was fermented. So any LTB that has growth and gas is considered positive, which we just talked about for the LTB. And you would need to take that sample and subculture, which means to basically take the bacteria from the LTB and use a loop using your aseptic technique, vortex your LTB to mix the bacteria, use your loop, dip it into the broth where the bacteria grew in the LTB, and then transfer that bacteria and inoculate a BGLB. And then you would take those BGLB tubes, put them in a can, and put them in the incubator. Now, one of the things that I want to point out, brilliant green lactose bile broth. If you remember what BGLB stands for, it literally tells you every important ingredient that's in the media, right? The brilliant green and the bile, those are the selective ingredients, and the lactose is the differential ingredient. So the name of the media actually tells you all the important things that are in there. So if you guys were doing this in class, you remember set up an unknown A and an unknown B, right? So we would have given you two unknown water samples. Unknown A was turbid with no gas. So unknown A in the LTB was turbid with no gas. So was A coliform? And the answer is no. That is negative for a coliform. It's a gram-negative bacteria, but it did not ferment lactose to produce acids and gas. So that's negative. And in fact, A in our experiment is actually Proteus mirabilis, which is gram-negative, but it's not a coliform. In tube B, unknown water sample B, we would have turbidity. It would be turbid with gas. So that would be our positive. And we would subculture to confirm. Oops. To confirm. So B would be the one that we would dip our loop into and inoculate a BGLB. A doesn't need to because A is already not showing it to be a coliform, so we would be done with A. But B, we would subculture. And if our water sample that we were testing, right, because these are just the ones we give you, the A and the B, if your water sample, let's say you tested your refrigerator water, if your water sample tested positive for LTB, then you would also inoculate a BGLB with your water sample as well. And so that is the second part of the multiple tube fermentation. So the last part of this is looking at day 3, which is the readout for the BGLB, basically to see if in fact we have coliforms. So again, the multiple tube fermentation, the purpose is to test a water sample for the presence of coliform bacteria, which is a confirmatory test. So for this day, you would be looking for if it had growth and gas, right? So if we see growth, turbidity, plus gas, that would be positive. That would be our confirmatory test to show that in fact it is a coliform because it's gram negative, which was what allows it to grow, and it fermented the lactose to produce the acids and gas. You can see here's our little air bubble inside the Durham tube. That's our positive. Again, our negative, just like for the LTB, our negative, there's two choices. Either we have no growth, or the second option to be negative would be growth with no gas. Because growth with no gas is still considered a negative because it is gram negative, but it did not ferment the lactose to produce the acids and gas. And so your readout would simply be to look at your BGLBs, look for turbidity, and look for gas. If you have turbidity plus gas, that is confirmatory for a coliform. And so this is just to show you. So E. coli was actually put in water sample B. So that unknown B was E. coli. And so this is the LTB that would result from E. coli. You can see that there's turbidity. There's a lot of cloudiness in here. And there's gas. Notice there's so much gas that the Durham tube started to float. You would have subcultured this one. So you would dip your loop into your LTB and inoculate an empty brilliant green lactose biobroth, BGLB. And what you would get is that with E. coli, because E. coli is a coliform, right? Notice again, we have turbidity and we have gas. You can see the little air bubble here in the Durham tube. And so if you see this, this would be confirmatory that in fact the water sample is or does include a coliform because we do see the acids in the endo agar and the gas in the multiple tube fermentation. And so putting all this together would allow you to see if the water sample you're testing, whether it be your refrigerator or bottled water or whatever sample you're testing, all of these tests combined would tell you if there are coliforms in that water sample.